All right. Test, test, test. I think my mic is, yeah, there we go. Perfect. I mean, I think in the UK they say it can be good or bad if all people know you. So we'll figure that one out later. So indeed, I'll share some of the studies that we've done in particular over the last 1.5 years since we used the Revio platform. And it really is in the context of rare diseases. But before I kick off, we have several collaborations with PacBio, and in some parts we even shared costs for some of the projects that I talk about. So take that as a full disclosure. I also wanted to start with a big shout out to the PacBio team. So since several years we're collaborating early on with Aaron Wenger, who's also here. That was a fantastic first partnership. We dared to do several trios with quite high coverage on SQL1. And oh boy, that was quite painful, but already along that way we learned a lot. And then a particular shout out to Mike Ebel and his great team. So rarely have I worked in the genomics business with a company with so excellent computational scientists, so that's a true privilege. And there's still so many win-wins along the way. So just to set the tone here for everyone, and I know there's some in the audience that are not working on human genetics and not on rare diseases. So rare diseases sounds always very exotic, and a lot of my friends and family ask, is it all worth to work on a disease where there's only a handful of patients around the globe? The trouble is, 7,000 probably is even the underestimate. There's newer numbers that may even suggest that there's 15,000 or so rare diseases around the globe. That makes it collectively not a rare disease. If you consider rare disease as an entity, it is a very common healthcare problem. It's estimated that 1 in 16 or 1 in 17 suffers from a rare disease. And for all of those rare diseases, there's high suspicion that a lot of them are due to genetics or heavily influenced by genetics. It's even estimated that 80% most likely have a monogenic Mendelian origin. Also important to stress, even in a very modern human genetics diagnostic laboratory, there is not this one single test that we apply to all the rare diseases yet. Even in Nijmegen, where I think we have a lot of automation, we have all really quite fancy workflows, even there we still have 15 or so test modalities, even for rare diseases. There's still these very difficult loci of the human genome where we still stick to old school tests. And potentially even most importantly, more than half of our patients, even with the latest tests, don't get an answer. And we need to change that, and we need to do better. The trouble with the human genome is there's a lot of variation that, of course, makes all of us unique and makes who we are. But the trouble for rare diseases, usually one of the 5 million or so variants that we carry in our genome can be sufficient to cause and explain a rare disease. So it's a very, very big haystack with a tiny needle that we have to find. I think Neil briefly mentioned that, of course, we're decent with genome sequencing. Imagine we use that as a first-tier test. We're quite decent on substitutions in particular. We're certainly much worse at indels and even much worse at repeat expansion disorders. There's a lot that we really do not get at all from short-read data. And we're even poorer at structure variant detection. And then you may say, but those 25,000 variants in comparison to the 4 million or so substitutions in the genome, who really cares? You should care if you are considered about the number of variant bases. The numbers affected by the different variant types are shown here. And it's quite striking that structure variants and insertions, deletions, and repeat expansions together explain more than 36% of genomic variation. And those are now exactly the variants that we're not good at by using our standard tools. To me, that is quite shocking, and that basically just means that we're still missing a lot of variation. Somebody sells us tools and methods claimed whole genome sequencing. They're really not whole genome sequencing if we have the ambition to look at all the variants. So that literally means for a lot of our patients, we're digging through that haystack, but that one needle may just not be in there, and that's very frustrating once you end at the bottom of the haystack and just realize a pretty obvious big needle was even not hidden in there. So I think, therefore, we need to drastically change our field. I think we, to some extent, failed, and we need a drastic change there. And that obviously is conceptually really sequencing in a better way. And of course, we all know there's, at this stage, at least 2 real long-read sequencing technologies around. There's some that claim that synthetic long-reads could also have some value. I have sincere doubts on that. So we early on thought we need to invest in real long-read technologies. And we have the privilege that we did so in Nijmegen. Since early days, SQL 1, then several SQL 2s, and now we have 4 Revios in our lab, and that's mainly what we'll focus on. And our ambition always is to use it for research, but then push really to first-tier clinical use. And really to do so and push forward, we need to basically usually do 3 studies. Ideally, you just go back to very difficult to target positive controls. If it's a new technology, you really want to understand that the difficult bits that your standard assays can find can be found back. The second important aspect is if you have patients that reach the end stage of standard of care technologies, how much extra is there to win? And then most importantly, to really launch a technology in first-tier diagnostics or in diagnostics in general, you need a prospectively designed clinical utility study to really make a strong case. Otherwise, you may well be maybe unconsciously biased, and you don't want that. So I'll guide you through those 3 steps of the procedure where we are right now. So, retrospective study of very difficult variants, and Neil showed that slide already briefly, but this all started on a study where my dear colleague Lysenka and her group, and the paper is published last year, asked the same question for short-read Illumina genomes. This is our current test portfolio, or was our test portfolio in 2022. We see around 25,000 patients with a suspected rare disease, and to offer all of the test portfolio to them, we need all of those different tests. Your test portfolio may look slightly different, but I think overall, it may not be so different. Maybe numbers differ, maybe 1 or 2 tests are not around in your lab, but I think that still represents even relatively modern human genetics departments. And if you then ask the simple question, can we just do Illumina genomes as a first-tier test and replace all of them? There's 2 answers to that test. The first one is nice. Illumina genomes are clearly the best first-tier test we ever had. They're clearly a little bit better than exomes. They are surely as good as exomes plus arrays together. So that's quite nice. But the inconvenient truth is also that there's hardly any test modality that we could completely stop. So while moving more and more from exome to genome sequencing as a first-tier test for most diseases, we still basically have to keep all the other test modalities alive. And that's, for some, they are not so cumbersome, but for some, you really need to continue to train people in-house, you need to write protocols, all of that nasty stuff, and we think that has to change. We can do better, but offer a better first-tier test. So basically, we went to that same study and asked our diagnostic experts cherry-pick those variants where the short reads really failed, or where we always needed another test on top, and that's what we did. So we went back to our diagnostic experts and told them from that respective study, give us 100 cases, 100 samples that carry jointly 145 variants that are extremely difficult. And there's no surprises really on such a list, so it's a lot of homologous sequences, it's a lot of repeat expansions, and there's details in those boxes. And then there's quite a lot of structure variants and complex aberrations, cytogenetic abnormalities, and then a tail of other variant types that are clinically important. And just to cut this story short, I was surprised. People that know me know that I'm the uber optimist. But by putting in these extremely challenging cases, I would have been happy if we hit whatever, 70% or so. No, we were much better. We hit 83% of variants that we automatically detect. So that means they're really called by the callers provided by PacBio. The ones that we add, it's 10% of variants. We need to develop our own tools or work a bit on visualization. So they're not perfect, but they're good enough to recognize the variant. So there's decent hope that you can, with further optimization, really automatically get also all of those. So jointly, that would be 93% of the worst mutations that we see in the clinic. But there's 2 particular aspects where we still have somewhat more of a systematic bias. One, and that's not unsurprisingly, I would say, are really complex cytogenetic aberrations. So that's also just showing you that we didn't cheat. We did not shy out from variants that we knew prior to the test we're unlikely to match here. So Robertsonian translocations were in there, or other repeat-mediated balanced translocations with breaks in or near the centromeres, for example. So that's not a surprise, and even for some of those, there may be a little bit of hope by utilizing pan-genomes or T2T and smarter tricks. Maybe there's indirect ways of calling some of those. Then, initially a little bit of a surprise, but I think by now there's a couple of other research papers out. While long reads are fantastic for repeat expansions in general, there's some exceptions to that with the current technology. So there's Canvas syndrome, RFC1 mutations, and Friedreich's ataxia. Those are extremely long GA stretches. And there, and that may be part of the mutational mechanism for the disease in first place, there the DNA may form so-called G quadruplexes. That means if we now utilize HiFi, where the polymerase reads the same molecule ideally 10 times, the polymerase literally bumps into a little DNA knob and can't continue. So it literally stalls or falls off or so. So most of those we still call in an indirect fashion, as a pseudo-deletion if you wish. So there's a hint for the variant, but of course, ideally we want to see the motif, we want to see the absolute size, we want to see the methylation. And we don't fully get that. Some we can recycle by going to the non-HiFi reads, and I think Mike and others are working on implementing that much more systematically, but it is so far a systematic issue. And we had one other issue, two cases. So those were females that carried a likely pathogenic variant and gave that to their son, and that was a diseased son, therefore. And we suspected skewing of X activation, and in two we didn't find it quite correctly. In fact, now the skewing tool that we built is much better than our standard of care tool. In fact, we believe now, I have quite strong evidence, that the initial two positive controls did not show any degree of real skewing. We just anticipated it was, but it was not. So it was 80 and 85%, which you could even doubt whether that really represents skewing. So there I think it's not a failure of PacBio or of the HiFi genome as a test, but rather the original standard of care test. We did one more thing with this data set, and that is asking the question, how much coverage do we need? So all of those genomes were done with 30x coverage on average, and the overall recall rate is already really, really good for all the automatically called variants, 96% and 20x, and close to 90% for 15-fold coverage. There's a subtle difference if you spread that out per variant type and per caller, so something like paraphase, where you have literally more than one allele, or something that is similar to another allele, there you still benefit a bit more from higher coverage. Potentially also any tools that utilize local or general assembly methods probably are a little bit better with higher coverage. But I think for us, at the moment, it's quite clear that 15 or even 20-fold coverage is already much, much better than anything with 30 or 40x Illumina coverage for the standard variant types for sure. So Neil alluded to that. This was published, and please read up the details. There's much more in the supplement. Previous slide. So of course, we're also aiming to use the multi-center approach to validate clinical utility for previously known mutations. So we have a deeper dive for some variant types, and we're working on the first manuscript. And there's several partners here in the room where we teamed up in what we call the HiFi Solves EMEA Group Consortium. So thanks for everyone's input. Friends from Uppsala and from Stockholm are here, and several friends from Belgium. So we're really teaming up with some strong clinical groups to provide a multi-center approach to validation of clinical utility. Then just two very brief vignettes on how much extra can we find. I won't bore you with that because the manuscript is just published, and Neil already had it on one slide. So we worked on the Solve4D consortium, but just as a reminder, that's pretty old data. It's actually HiFi sequel 2 data with just 10-fold coverage. And even there we found not only structure variants, what we designed the study for, but we also found repeat expansions quite easily. With 10-fold coverage, you really get some positive calls quite easily, and even some point mutations, even with 10-fold coverage. And we solved 12.6% and generated new candidates and several. Those are largely usually structure variants, but the trouble, different from point mutation, structure variants are quite often private. So you see them for the very first time. So it's a bit more laborious to make the case that they are disease-causing if they don't immediately hit a gene that you know and where you know the disease mechanism. Since ever then, we moved on. We've done 500 additional samples to answer the question about extra yield. But I just summarized that on one slide. Sorry for being brief, because I have one more study to come right after. This replicates what the Solve4D study has shown and several other global studies. Somehow the extra yield for now seems to be 10 to 15%, but let's be honest, that's still what I consider the low-hanging fruit. Those are the variants that quite easily jump out of those kind of studies, may it be trios, may it be unsolved families, or even singletons. I think there's even much more in the data that we don't understand yet, so there's potential for even much higher yield. But now I want to switch gear to our very latest study, and that is a prospectively designed clinical utility study to really provide evidence that we can use this in the clinic as first-tier and generic test. And to do so, we did a pretty large study by just mimicking standard of care. So on the left side of that decision tree, you still see our standard of care. Be reminded, those are more than 10 different test modality. So that's mimicking the original Illumina study. We did 1,000 genomes. And again, we did 1,000 genomes here, went to the year 2022. The benefit of that is that diagnostic standard of care is completely finished. And we can even also ask the question, the same sample that was tested in 2022 and was either positive or negative may have had other tests the year before or the year after, so we can even project the data. So Lysenko and her team, they again went back to 2022, selected 1,000 representative samples that represent our diagnostic referrals, our diagnostic yield, and the use of workflows. And then we asked 3 of our postdocs to really do a systematic assessment of that. But they were no experts in diagnostics, nor were they known to the outcome of standard of care, so they're completely blinded. And to their big delight, it all works. Nobody got fired from their team. Even non-diagnostic experts can do now diagnostics on these data. And the way this works, this is the cohort assembly. So the most important takeaway is this really mimics diagnostics of more than 300 different clinical indications. It's representative for trios, for singletons, and on the lower right, you see all the representative workflows that our standard of care offered in 2022. But if you want to do that on 1,000 samples moving forward on our 4 Revio systems, we decided we can't do that just by manual library prep. So there, our fantastic team in our department optimized a fully automated library prep with some early collaborations with PacBio. Then we did HiFi sequencing again with 30-fold coverage, and we utilized bioinformatic analysis suite, largely using the callers from PacBio, and then there is a little bit of other colors in there. It's usually just in-house annotation tools that we used. If you're really interested in all the QC of such a cohort of 1,000 samples in the automated library prep, refer you to the workshop that our wonderful research technicians, diagnostic technicians, I should say, Ronnie and Amber are doing. They're really global experts for PacBio libraries since many years, and they really managed to get excellent libraries. And in honesty, one more remark on that. If you would have asked me 2 years ago, can we automate long-read libraries completely on the Hamilton robots? I was a bit skeptical, but to my surprise, because we were quite spoiled by very careful size-selected gel extraction methods, this all worked, and it even worked on bank material from 2022, and some samples were much older than that. But again, Ronnie and Amber will give you all the insights into how 1,000 real-life samples look, and I'm honestly really surprised that we can now scale that, and it's not the rate-limiting step anymore. We can just update. We have 2 of those robots now. They can currently do 24 samples in a run, soon 48, and we can even upgrade to 96. So it's really just not the bottleneck anymore. Quite a surprise, and I wouldn't have signed for that a year or 2 years ago. Of course, there's a concern in terms of how do we do the analysis. In our house, we're extremely pleased that Christian Gillison is heading a team of more than 30 bioinformaticians. In this case, Raoul and Timon in particular just morphed our graphical user interface that our diagnostic experts already use for exome and genome interpretation. So in fact, all our diagnostic staff members don't need to be retrained. They already know that platform. We just squeeze in other data. So in effect, they don't even know that we now offer, or start offering soon, HiFi genomes instead of Illumina genomes or exomes. It's only a few subtle differences. So what we still do here, we see the sample ID, very easy. You can select a gene or a gene panel. So that's what we did also in this study. If there was a request just for hereditary breast cancer, we only look at the handful of genes for hereditary breast cancer. We don't look genome-wide, of course. Then we have a tab per variant, and that's really where the only change has happened. Now we have a few tabs for structure variants, for STRs, and for paraphase, so called and paralogous gene sequences that don't exist for other short-read data. That's the only add-on to the entire software. And then you can go for some pre-selected filter or preset filters, but you can also customize them if you want. That's exactly how our diagnostics is run since years. And then you see a little overview of how many variants are left. And in this case, we indeed sequence the family with a suspicion of a familial breast cancer. And after all filtering, there's a single check2 variant left that is known to be pathogenic. And then you can just report. So positive controls, extremely simple. You just click, tick, 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 and you're done. So now I'll just go to the next slide. Let's see. Yes, so this is a preliminary overview. We're still finalizing the analysis, and we're starting to write up that study. But this is for 780 index cases, the comparison of standard of care to HiFi genomes only. So on the lower left, you see the outcome of standard of care. There, our diagnostic yield is only 15%, and a very likely VUS plus in 12%. That's sometimes shocking, so that is not even close to 30% if you sum that up. But in fact, nowadays in diagnostics, we offer a lot of genetic tests also to exclude a genetic diagnosis. So there is always a large chunk of negatives, at least in our center. And I'm sure that's true for several of your centers. And while that may be a surprise, extremely good news is that in the lower right corner of that box, 564 samples are negative by standard of care, but they're also negative by HiFi sequencing only. While you would have hoped that 200 of those are now positive and get a proper diagnosis, this is extremely good news. Because the most scary thing that could have happened is that 200 of those end up as an uncertain variant, or that you need 2 weeks per case to go through the new data. Imagine you have a technology that delivers a lot of false positives in the panels that you study, you have a major issue. In fact, you would just kill your entire diagnostic workflow. So that is not happening. The data is extremely clean, and even non-diagnostic experts can now click 3 times and we get very similar results. So our negatives stay negative and are easy to analyze. That's extremely important. But maybe indeed more interesting, we missed some variants. 6 cases that got a positive result from standard of care were now negative. 5 of which were simple annotation mistakes or interpretation mistakes. Those were variants in the UTR that we were not aware are pathogenic, so the diagnostic expert working on that one gene or that one disease would have easily scored that. And in fact, we improved our annotation, so that's solved for all 5 of them. So those are nasty little mistakes of using a research team mimicking diagnostics. There's one that was properly due to coverage issues, so that was one of the samples where we had lowest coverage, 16-fold genome-wide coverage, and by chance the disease-causing mutation only had 2 variant reads and was not apparent. We had one other technical issue, so 2 out of 1,000 samples had an apparent technical issue that was related to the sequencing. There was one mapping issue for a variant of so a VUS in the standard of care that was now negative, so we don't even know whether that impacted the exact diagnosis. There were 2 cases that had a likely diagnosis based off standard of care. So that was, I think, exome sequencing. We found 2 hits in a recessive gene. And then the report said it's a likely VUS, those 2 variants, but we can only make a firm diagnosis if we get parental material to do segregation. That segregation had not happened, so therefore it stayed a VUS. But now with the HiFi data, we can directly phase and tell, actually, this is not the disease cause because it's 2 rare variants in a recessive gene that may fit the phenotype, but they're in fact in cis and not in trans. So they are usually, I always thought we have so good connections to our clinicians, to our families, we should always get DNA samples within 1 or 2 weeks after the test result. That's really not the case. Sometimes that can take years. And now we don't need that. We can directly phase in the data. And we see a very similar effect for the ones that were moved up from likely diagnosis to certain diagnosis. There's 23 in there where phasing indeed provided the evidence that 2 recessive hits were in trans and therefore explain the disease fully. This is not rocket science, but it is extremely convenient in the clinical setting. And there were several cases in total where we found new variants that the initial test had clearly missed. And I'll just show you one of that, I think, on the next slide. Let's see what that is. There we go. So here we had a Marfan type of phenotype. Bart Lewis is working for 10% in Nijmegen, and he clearly got the result back. The standard of care found a VUS in FBN2, where he clearly, after unblinding, we saw that he made the record. A VUS and FBN1 would have been a much better fit, so please find a variant in that gene. And now we did. There is an intergenic tandem duplication that was initially missed by exome sequencing. One more slide, or I think 3 little vignettes, and then I'm done. I told you that phasing is very important, and no surprise, with the long reads, you can phase much better than with short reads. In fact, looked at all the relevant disease genes. We always have very high likelihood. Of course, there can be sometimes samples that have non-informative SNP markers for a longer stretch. For example, if there's large degree of inbreeding, you may have an issue, but otherwise you can always phase your data. So that's really convenient for the clinical setup. We also looked at methylation, so you get that now for free, where previously we had to do separate tests usually. And in this study, a positive control that scored positive in standard of care and the HiFi sequencing was a case with Wiedemann-Steiner syndrome. We found a quite convincing loss of function de novo heterozygous mutation. But for that gene, an episignature, or in fact, 3 episignatures have been published by other groups. So we just pour out those individual CPGs from the methylation array that called that episignature, and just a simple z-score analysis already showed that that same sample here in the red square is really giving us the highest score for those 3 published episignatures. So even on a single sample basis, you already now get a molecular phenotype. Now you could say for a stop mutation, it's not so spectacular. I don't desperately need that signature because it's clearly a pathogenic mutation. Imagine that would have been a missense that is private. This really would be the molecular phenotype to make a stronger case. We're just not used to it, that you get it from the same assay, so that's very convenient. We were curious, there was a second sample, the green dot in there, that also scored almost as high. So we asked, could that also be a case with Wiedemann-Steiner that we didn't diagnose? No, in fact, that is samples with Down syndrome, and they score even higher on the published Down syndrome episignature. So no surprise there, and therefore they have this bit of a score also in the other signature. So I think we're utilizing that more and more for all the previously published epi-signatures, but of course we can build signatures from scratch, and that's what Christian's team is doing with Michel in particular. One small the last vignette for this is, if we have these new data types, we may also need new ways of analyzing them, and we had the privilege to already host 3 hackathons or solvathons. So we brought together clinicians, in some events even patients that showed up in Nijmegen, and brought together experts from around the globe to really look at these data jointly. And it really works. Within 2 days of having long-read data on the table, you crack cases and give a diagnosis to unsolved cases. We did this with the wonderful Wilhelm Foundation. They had the first edition in Sweden 2 years ago, and then last year we hosted the undiagnosed hackathon in Nijmegen. Just a wonderful event and community effort. We did something similar in Solv4D. In fact, the paper I've shown, part of the findings came out of the long-read solvathon. And just 2 weeks ago, everybody in Nijmegen and Maastricht came and said, hey, you do these global events on solvathons and hackathons, we want one in-house. So we provided 50 long-read genomes for previously undiagnosed cases, brought 100 people from Nijmegen and Maastricht together in the same room, and we cracked directly 5 out of 50, so 10% extra yield by just looking at the data with all the experts in the room. So that's not only fun, but it really works, and it educates people to get used to these new data types. There's a few remaining challenges, or opportunities, I would rather say. My prediction is that there's many unsolved, undiagnosed rare disease cases where we will make discoveries of previously missed repeat expansions and structure variants. I think we will really embrace much more the value of methylation as a molecular phenotype that's readily available now in the data. I think previously by doing separated arrays or bisulfite nasty sequencing, none of that was really flying in the rare disease field. I think that's now really nice to have baked in. I think, and that's much more of a conceptual concern, only once we read genomes to completion, we can start tackling non-penetrance or reduced expressivity of disease, or even discover novel genetic modifiers. That does not work if you don't look at the entire genome of a patient. My prediction also is that we will start finding many more non-coding mutations, and in my prediction, they are largely enriched for structure variants. It's much easier to destroy the function of an answer by completely removing it or moving it elsewhere than just a single point mutation. So I think that is a flaw of previous experimental designs. Now we can really do that. Maybe that also speaks again to my optimism, but I think again, by only sequencing complete or near complete genomes now, we will get better and better at interpretation. And that together with all these wonderful assays, saturation genome editing and other massively parallel functional assays, together with AI predictions, I think will solve the VUS problem, at least for the classical variant types, to a large extent. I bet in 5 years from now, we will have still some VUSs, but not as significantly as we have right now. And again, these complete genomes will help massively. So that really brings me to the end of my talk. I hope I gave you first glimpses that in our view, HiFi long-read genomes are very comprehensive genomes. We start understanding that methylation has great value. Important to stress, structure variants and STRs are largely overlooked by short-read methods. For now, rare disease studies, several of those, also by others, indicate 10 to 15% extra yield. Everybody who's in the rare disease field knows that's quite massive. And if I'm excited about that, that's one thing, but luckily also all management is excited enough that they just purchased 5,000 HiFi genomes to go live in diagnostics this summer. So that's what we're working for. I think more conceptually, so bring that message back home. Human genetics is not only fun, but it's even getting easier and better from now onwards. We, for the first time, can really lead near-complete genomes. So we can forget about talking individual variants. We can really start talking about alleles and haplotypes, can embrace phasing much more than ever before. And one last more conceptual part, for the first time we may approach genetics, rare disease genomics, in a Bayesian approach. For the first time we can say, hey, we have this one missense variant, which clearly is a variant of unknown significance. But if we finally read the rest of the genome, we have convincing evidence that there's nothing else that makes sense in light of the phenotype of the patient. We may even upgrade that variant of unknown significance. ACMG already has that in its guidelines. And of course, now the work is up to many of us, hopefully also jointly. We will scale this, we will work in consortia to provide even more clinical evidence, but we'll surely go live this summer, and we think we'll do a much better job for our patients. There's many patients to thank. Of course, also many patients to thank, but also many employees to thank, both in Maastricht and in Nijmegen. Ronnie and Amba are here. They're really the technical experts for anything library and machine related, and they will give a wonderful talk tomorrow about that, our workshop. But there's also many colleagues and collaborators at PacBio with whom we collaborated over the last year. So thanks a lot for that. And a final plea, I'll just invite all of you to join us in Milan for the science, but maybe also for the gelato and some chats. So thank you very much for your attention.